for over 2 decades, what we all called the complete human genome, well, it was anything but. We had our own biological instruction manual, and a huge chunk of it was just blank.
That's right. For 20 years, 8% of our genetic blueprint was completely missing. An entire eighth of our DNA map was just a void, a question mark. So, what was hiding in those blank spots? And why on earth did it take so long for us to finally read it? Well, the answer is a fascinating story of a puzzle that seemed impossible and the incredible tech that finally solved it. To really get what we found, first we need to look back at the map we used to have. The original human reference genome, which scientists call GRCH38. You can think of it as the master blueprint for a human being. It was first drafted way back in 2000, and it became the go-to guide for researchers everywhere. It was, without a doubt, a monumental achievement, but it had some pretty big holes. I mean, can you believe that? This foundational text of our biology had over 150 million characters, just replaced with placeholder letters? We're not talking about a few typos here. These were entire sections, whole chapters that were totally blank. And this wasn't just random filler DNA. We were missing centromeres, which are absolutely critical for our cells to divide correctly. We were missing the genetic factories that build proteins. And we were missing these huge, duplicated regions that are hotspots for evolution and disease. These were fundamental parts of our biology, and they were completely invisible to us. So the crucial question is why? What made these parts so impossible to read for so long? What made this 8% so stubbornly difficult to figure out? The problem was repetition. It's been perfectly described as trying to solve a jigsaw puzzle where most of the pieces are just identical patches of blue sky. You see, vast stretches of our DNA are made of the exact same genetic sequence repeated over and over and over again. See, the early sequencing technology could only read tiny little snippets of DNA at a time, just a few hundred letters. So when it hit a repetitive sequence, thousands of letters long, it was totally lost. It was like trying to assemble that blue sky puzzle with a million tiny identical pieces. You'd have no idea how they fit together or even how many pieces there were. It was basically impossible. So, to solve this puzzle, a global team of scientists, they called themselves the Telomere-to-Telomere, or T2T Consortium, needed a massive technological leap forward. They needed a way to read not just genetic letters, but entire paragraphs at a time. And they got it. They combined two incredibly powerful new technologies. One, called PacBio, was like a super high-resolution camera, getting really accurate reads of long DNA segments. The other, Oxford Nanopore, could read these ultra-long stretches. We're talking over 100,000 letters at once. Together, they could finally read straight through those repetitive sections and see the whole picture for the very first time. And the result of all their hard work is just a monumental scientific achievement. It's a new, truly complete human reference genome called TT-CHM13. And just look at the sheer scale of this improvement. It's night and day. The number of gaps, it went from over 120 million missing letters down to 0. The number of separate pieces the genome was in, it went from almost 1,000 fragments down to just 24 seamless sequences, one for each chromosome. This is a quantum leap in quality. And there it is, the grand total, 3.055 billion DNA base pairs. This represents the first ever, truly complete gapless sequence of a human genome. This chart illustrates the journey so perfectly. You can see how for over a decade, the completeness of the human genome barely budged. And then, bam, that massive jump in 2021. That's the T2T Consortium finally completing a 20-year scientific quest.
Okay, so this is the big reveal, right? We've filled in the map. What treasures were actually hiding in that mysterious 8% this whole time? Well, it turns out the vast majority of it is that super repetitive satellite DNA that we find in centromeres, the very regions that were so impossible to assemble before. This confirms that these incredibly complex, repetitive structures are a huge part of what makes our genome tick. But it wasn't just repetitive stuff. Buried within all that new DNA, scientists found predictions for almost 2,000 new genes that we never, ever knew existed. And get this, Of those, at least 99 are predicted to code for brand new proteins. That means we've basically discovered new machinery inside our own cells. These could be the keys to understanding immune responses we couldn't explain before, or developmental processes that were a total mystery. It's like finding a whole new set of tools in our body's toolbox. And this is the real takeaway. Unlocking these previously hidden regions opens up entirely new avenues for research. We can now accurately study how these complex areas contribute to our evolution, to the incredible diversity across our species, and to diseases like cancer, where things often go wrong in these exact same regions. So, this isn't the end of the story, not even close. It's the beginning of a whole new chapter in genomics. Having the full, complete reference book lets us explore our own biology with a clarity we've never had before. The real discoveries? They're just getting started. Thanks for joining this explainer.